For the diagnosis, the first sample that I will take will be a stool sample, and the clinical rule is that I have to take a 3 consecutive day stool sample before I rule out this. So 3 days consecutively, ask the patient to get his diarrhea sample to your lab. In the stool sample, either you can see the trophozoite of this or you can see the cyst form. When I see the trophozoite, I can very well notice that there is red blood cells being eaten up. I know that is erythrophagocytosis. And it has how many nuclei? I can see only one nucleus which has a central dot known as central karyosome. So the nucleus with that central dot gives a cartwheel appearance. These two buzzwords are important. The trophozoite shows you erythrophagocytosis and cartwheel appearance. Very, very important. That's the trophozoite. Now let me talk about the cyst form, in which I can see anywhere between 1 to 4 nuclei. I can notice in this I have the 4 nuclei stage, and there is a very, very thick body which is known as the thick chromatoid body. That makes a cyst form. Let me show you real pictures. This over here, if you notice, is the red blood cell eaten up, and here you can see a nucleus with a central dot which is the cartwheel appearance. So this is the trophozoite. Now when I show you the next picture over here, I can again see only one nucleus. I thought that okay, trophozoite also has one. The cyst has anywhere between 1 to 4, but this has a thick chromatoid body or bar, so this has to be the cyst form. This is Entamoeba histolytica. If you give this diagnosis, the clinician will start treatment, which I will tell you in a while. But sometimes in the stool sample, I also get to see Entamoeba coli. Before I explain that to you, this is a normal parasite which can be found in anyone's intestine normally and can be found in anyone's stool sample also normally. So if this is found, don't start giving unnecessary drugs to the person. How does Entamoeba coli look like? Again, it has a trophozoite form and a cyst form, and keep comparing it with Entamoeba histolytica. In Entamoeba histolytica, you had a central karyosome. So in Entamoeba coli, you have a karyosome at the eccentric, a little periphery, so it will not show you the cartwheel appearance. How many nuclei? Again, it has only one nucleus. That's the trophozoite. But when I come to the cyst has anywhere between 1 to 8 nuclei. You can count in so many pictures that I have put up over here. It has 8 nuclei, and the chromatoid bars are also very, very thin out here. I just revised the nuclei with you. In Entamoeba histolytica, trophozoite versus cyst. Trophozoite had 1 nucleus, cyst had 1 to 4. In Entamoeba coli, the trophozoite has 1 nucleus and the cyst has 1 to 8. My question here is that this chromatoid body, be it thick or thin, what is it? What is the composition of a chromatoid body? And the options will write RNP, ribonucleoprotein is what it is made up of. Now a question for the INI-CET students is, what is the special stain of a chromatoid body? It is iron hematoxylin, which you have to mark. Now coming back, all of this was when you had a stool sample, or basically patient having diarrhea. Let's assume patient has something to do with liver involvement, anchovy sauce appearance. I got the brown color pus. When I look at it under the microscope, you know you will always see the trophozoites with erythrophagocytosis. Whatever may be the organ, ultimately you will do the culture. So if there's a stool sample, I do a stool culture, and these are the 2 sample culture media that I can use. Get used to their names. When I say axenic culture media, A means absent, means it's only culture media. It has pure culture media. It has no random supplements added to it. But when I use the word polyxenic culture media, it has a lot of supplements, like bacterial supplements have been added to it. So now out of these, the axenic culture media has only one example, that is the diamond media. How I have learnt it is, diamonds are pure and axenic is also meaning a pure media with no supplements, because the rest of the list is very difficult. That's why I just learned diamond as pure, and rest everything else goes under the polyxenic culture media. So when they talk about Nelson, Balamuthia media, Bock and Derbolov's media, Craigie's media, all of these are polyxenic, so I don't learn it. I learned diamond is pure, axenic is pure, so this combination is easier. Finally, let's treat the patient. It's a case of diarrhea. I will be giving metronidazole for 10 days, but the story doesn't end there. Read option A will be metronidazole, option B will be metronidazole and paromomycin, which is the correct answer, because metronidazole will only control the diarrhea. But what about the amoeba which is living in the intestine and the lumen of the intestine? You have to go and kill it over there. So luminal amoebicide is paromomycin. So metronidazole followed by paromomycin is given. So you're done with the first set, and that is Entamoeba histolytica. The next family of amoeba is free living, means they are freely available in the environment, in the soil, the water, the air. And there are 2 organisms that come under it. One is Naegleria fowleri and the other is Acanthamoeba. Let's start with Naegleria fowleri. Both both of them are categorized many a times by news channels and a lot of headlines as the brain-eating amoeba because they will attack the brain. Naegleria fowleri causes something called PAM, primary first-time infection, amoebic because it's a free-living amoeba, meningoencephalitis because it's a brain disorder. Whenever I say primary, it's happening for the first time, so its clinical course is going to be very acute. In fact, it is so fast that many a times they will say that patient has died within 5 days. It just happened, meningitis happened, 5 days patient has died. It can be fatal very, very fast. So now my question is that how is this deadly disease happening? Usually by swimming in dirty water. They don't give you this history. They say patient has gone to his village for a vacation, or during school days, vacation, summer vacation, the student has gone to his village. Now you assume that in the village he or she must have taken bath in a dirty pond and dirty water. So clearly the examiner has not visited villages and not known that nowadays we have so many facilities there. But nonetheless, still the examiner mentions the word village and you assume that swimming in dirty water goes hand in hand. Now whenever we swim, let me show you a picture. What happens is that the trophozoite form of Naegleria with that swimming water enters through our nose. From the nose, what is that plate over here? Cribriform plate. From the nose, it goes through the cribriform plate and reaches our brain, and in the brain causes PAM disease, which I just told you. So repeating, the route with which it is going to spread is going to be the water going through the nose. What is going through the nose? Which form is the infective form? It's the trophozoite. From the nose, it will go to the cribriform plate, and through the cribriform plate, it will go to the brain and cause the disease PAM. There's an easier way of learning it, which learns like, it's like a song that I'm making you learn, that when you're dealing with Naegleria, they will give you a history of Nale mein nahana. So taking bath in dirty water, the water will enter through your naak, that is nose, and will go to your brain, that is neural. So if they give you a history of Nale mein nahana, naak, neural, you know you're dealing with Naegleria. Let's talk about the other one, that is Acanthamoeba, which is also brain eating, and it causes granulomatous amoebic encephalitis. So encephalitis is brain, amoebic, that's an amoeba, granuloma, it causes a granuloma in the brain. Let me go back to pathology chapter 2. Granuloma is acute inflammation or chronic inflammation? Of course, it's chronic inflammation. So here, the course is going to be chronic. But apart from that, it also, apart from the brain, it causes involvement of eyes. Contact lens keratitis is also noted. And do you know this is seen in HIV positive or immunodeficient individuals? And I have learned that Acanthamoeba, answer is always all of the above. Whenever they give me options, I will say all of the above. So if they ask me, what is infective? Cyst is infective or trophozoite is infective? I'll say both, all of the above, all are infective. Then which route does it enter my body? Does it come through the eyes? Does it come through the nose? Does it come through the mouth? And I'll say again all of the above. Whichever route it has entered, eyes, nose, mouth, ultimately it will go into the blood and it will reach my brain. So can I say that Naegleria had that neural route through the nose going direct to the neural route, whereas Acanthamoeba goes and travels in the blood and then reaches the brain? Let me show you a picture here. Over here in Acanthamoeba, the cyst form can also come to us. The trophozoite form can also come to us. So both of them are infective. It can come through the eyes, through the nose, through the oral cavity. So all of the above. This is the trophozoite over here and this is the cyst. How do they look under the microscope? Are previous year questions. Very, very important. Listen carefully. When I say acantho word, it means spikes. So here you can see the trophozoite, if I zoom in, has these spiky porcupine cactus-like projections coming out. All spiky things coming out, that's the trophozoite. Versus when I look at the cyst, I can see there are two walls and both the walls are very zigzag, basically wrinkled. So the cyst is going to have a wrinkled double wall, and that is how you differentiate. And if you've learned up till here, I'll just change one thing in the story and a new organism will be ready. There is, what did you study right now? You studied Acanthamoeba. I'm just going to compare it with another amoeba called Balamuthia mandrillaris. So what did you study right now? That Acanthamoeba causes granulomatous amoebic encephalitis, plus it causes the eye involvement, contact lens keratitis. But Balamuthia only causes granulomatous amoebic encephalitis. It does not cause any of the eye involvement. That's feature number one, difference number one. Difference number two, in Acanthamoeba, I studied it is only going to cause infection when the immunity is down, immunodeficient HIV. Balamuthia can cause infection in both situations, immunity down, immunity normal, it can always cause infection. Then Acanthamoeba, I studied that the trophozoite is going to have spikes and the cyst, if I can draw it, is going to have a wrinkled wall. In Balamuthia, it's the opposite. The trophozoite has a classical something looking like a fish. It's a fish-shaped trophozoite trophozoite which they have, and the cyst has a very, very smooth wall. So overall, if I had that one buzzword for Balamuthia, it would have definitely been the fish-shaped trophozoite that it has. Finally, all your free-living amoeba are almost done, but now out of whether I say Naegleria or Acanthamoeba or Balamuthia, any free-living amoeba, the culture media for all of them is the same. NNA with E. coli. What is NNA? No one's asking you. Just go through the full form. It is a non-nutrient agar. What if they write the full form in the exam? You should be able to decipher it as NNA. So non-nutrient agar is what is used. But gold standard will always be genetics for the diagnosis. So at any point they mention nucleic acid amplification testing, that is going to be the gold standard. All the free living amoeba are done, and we come down to my favorite amoeba, which is the easiest to learn, and that is going to be Balantidium coli, because if you know the name, you know everything about it. When I say Balantidium coli, coli word will be read as coli. So this is the only ciliated protozoa which is going in the intestine. If I show you this first picture over here, you will easily be able to see how these hair-like cilia are coming out of it. So the only ciliated protozoa is going to be Balantidium coli. So coli, cili is okay. Now let me come to Balantidium B. Everything now follows B. If I ask you what is the size, you'll say, oh, it's one of the biggest. It's the largest protozoa that we are dealing with. Okay, so which part of the intestine does it go to? B for big. It goes to the large intestine, and it is causing diarrhea. So I have two questions now. How did I get this infection? Because again, we are having diarrhea. So of course, there must have been some dirty food and water, street food that we must have eaten. So which is the form that is infective to me? It is going to be the cyst form which is infective. Now let's continue the story that when this patient will have diarrhea, I want to finish off the pharmacology. I will be treating it with doxycycline. But before that, make the diagnosis. Patient comes with diarrhea, look at it under the microscope. I will see again trophozoite and cyst form, and both of these are binucleated, means trophozoite also has 2 nuclei, cyst also has 2 nuclei, so another B coming my way. So this is the biggest protozoa. It involves the big intestine or the large intestine. All the forms are B for binucleated. So I can see the 2 nuclei over here. The bigger one, what is the shape? B for big, B for bean shape. The big nucleus is bean shape. The small nucleus is a dot. Let's write it down. There are 2 nuclei. The bigger or the macronucleus is going to be bean shape, and the small micronucleus is just like a dot. So a quick recap, Balantidium coli is the only ciliated protozoa. It is the biggest protozoa. It is involving the big or the large intestine. It is always binucleated, whether trophozoite or cyst. In those binucleation, the bigger nucleus is going to be bean-shaped. Treatment is with doxycycline.